We'll begin with the immune system physiology and look at innate defenses, where this is the body's ability to defend against foreign invaders. The immune system is a little different, where it's a functional system rather than an organ system. And it's broken down into two different categories of innate defenses and adaptive defenses. So the first one that we will start with are innate defenses. And so these are going to be defenses that include surface barriers and look at internal defenses. So surface barriers, the biggest one is going to be our skin, but also we have different mucous membrane that secrete mucus and are aid in getting rid of different foreign objects within the body. We also have specialized cells such as phagocytes, natural killer cells that help with getting rid of unwanted materials in our body. But also we have these different responses like the fever, which is important in combating different types of foreign molecules. We also have antimicrobial proteins that are inert and that we're born with, and also inflammation, which is actually an important response for homeostasis. The second part are adaptive defenses, and so this includes the blood, and so looking at B cells, and then also looking at within different types of cells with our different T cells. With innate immunity, what we have are structural defenses, and so these respond to nonspecific foreign substances. And so we have a first line of defense against which pathogens are exposed to the barriers offered by different surfaces that are exposed to our external environment. However, though, this is not an immune response because this is actually a physical barrier where very few pathogens can penetrate our skin. Other areas that we have include the hair, at the entrance of the nose, and then also coughing and sneezing reflexes to help keep pathogens out of our bodies. And with our mucous membranes, we have mucus that's secreted by the epithelial lining of the respiratory tract, the upper GI, that has these antimicrobial chemicals in them. And so they make mucus that is sticky, where different particles will stick to them, and then they're prevented from entering the blood. Also, we have cilia in certain areas that help to move dust and debris along so that they are swallowed instead of entering into the bloodstream as well. The second line of defense that we have is the inflammatory process, and this is usually in response locally to some type of infection or injury. And so the purpose of inflammation is to destroy or inactivate foreign invaders and to set them up for, or set up the stage then for tissue repair. And so one of the key players in this are going to be phagocytes, and these are important because they're the ones that are responsible for degrading a lot of the different pathogens. And so these include neutrophils, macrophages, and dendritic cells. Adaptive responses are going to be in regards to specific foreign substances. And so we have two different types where within our blood we have different types of B cells, which the B cells are responsible for creating different antibodies, and so they can bind to specific antigens and then get rid of the foreign substance when we have that antigen antibody binding. And then we also have our T cells, which recognize specific antigens, and they also can induce different macrophages to help and get rid of different types of pathogens, so such as like cancer cells as well. When we look at the first line of defense for the innate immune system, let's begin with the skin, where skin is the physical barrier to penetration by different pathogens. And it also has lots of different secretions with lysozymes that are enzymes that are responsible for degrading bacteria. Also, there is keratin, which is water insoluble protein that is formed, and so this helps to resist penetration of bacteria and toxins into our skin. And also the lysozymes that are produced have a secretion of a low pH, and so we'll see this again where these acidic environments help to degrade the bacteria and break down their cell walls. Mucosa also serves as a physical barrier, and it produces a variety of different protective chemicals. And this also is important because it allows the prevention of these pathogens from entering our bloodstream. We find gastric mucosa specifically within the GI tract, and so this is very high in acid, and especially within the stomach, very low pH, and helps with production of different types of proteolytic enzymes, which break down different types of proteins found within bacteria. We also have saliva and lacrimal fluid that have lysozymes. So for our eyes, we constantly are being flushed with the lacrimal fluid, so that prevents bacteria from entering into the nervous system as well as within the bloodstream. And lastly, we have mucus, and so this is important for trapping and moving epithelial or pathogens away from epithelial surfaces, and usually this is accompanied by cilia to help with that movement. And so we find that with the respiratory tract and also with the urinary tract as well a little bit, and with the reproductive tract, especially with females as well. So in this picture, what we're looking at is the plasma membrane, and what actually detects the different substances as being foreign or not are going to be these glycocalyx. And so these are going to be extensions of carbohydrates that come off of proteins. And so here the purple is representing a protein, and then the green represents just another carbohydrate. And so again, cells don't have eyes, they can't walk around, right? And so this is one way that they can communicate to each other is by recognizing different signals, and if they are going to be foreign or not based on these carbohydrates that are attached to the proteins. And so this is important again for if it recognizes a pathogen that's going to recognize as itself, or if it's another cell similar to itself, and so it's not going to induce any type of harm. So phagocytes will be the predominant cell type that we will see within the innate immune system. And so a phagocyte is a cell, a type of cell within the body that's capable of engulfing and absorbing bacteria and other small cells and particles. And we have different types of phagocytes that we find throughout the body based on their location. So the first are macrophages, and so macrophages are derived from monocytes in the blood, and we have two different types. We have free and fixed, and these are either going to be roaming through tissues, such as with the free macrophages, and so they're always scavenging, always looking for different types of bacteria that it can get rid of. Or we have fixed macrophages that stay stationary in certain areas, so such as like within the liver we have Kupffer cells, and then within the brain we have microglial cells. So that with these fixed cells, what happens is that the blood that flows through like the different areas will be phagocytized by either the Kupffer cells or the microglial cells. And so, but all of them have the same function and role where they're going to get rid of cellular debris, look for different pathogens such as bacteria and fungi, and then they'll have lysosomes that are going to be embedded with the vacuoles to get rid of that foreign or debris particle. So neutrophils, which again are going to be the predominant white blood cell type that we have, are going to be responsible for phagocytosis. And so part of the reason why they have that, they're granular, is because they have chemicals within those granular dots within their cytoplasm, and so those are actually chemicals that are involved in vasodilation and also with degradation of different phagocytes, and so those are different enzymes too that are responsible for that degradation. Also in fewer number, but we have this, are eosinophils, and these are responsible for attacking parasites, again, are a type of white blood cell. And they also participate in hypersensitivity to different types of reactions that we have. And the last type of phagocytic cell that we have are mast cells, and these are found around connective tissue and are important because they also are have a phagocytic ability to get rid of bacteria. And a big thing with them is that they also release histamine and other chemicals that are involved in the inflammatory process. So the way that the different phagocytes work in terms of killing different types of pathogens like bacteria is that there has to be first contact between the surfaces of the phagocyte and the microbe. And then this is going to trigger the phagocytosis during the contact of the interaction between the receptors and that glycocalyx of those carbohydrates, or sometimes of different lipids in the microbial cell walls. However, sometimes there might be a coating of like a thick gelatinous coating around the microbe, and so that's sometimes not enough for our phagocytes to detect the different types of carbohydrates or lipids. And so a chemical reaction has to occur where this is opsonization. And this is due to the substance that is called opsin, from the Greek word of preparing to eat. And so this helps to mark and enhance the phagocytes. Once we have the endocytosis of the microbe into that phagocyte, then a phagosome forms. And so these are respiratory bursts where the merging of the phagosome then is going to merge with lysosomes, all right? And so from there, there's going to be free radicals that are going to start degrading that pathogen. And so also there's going to be different types of proteins called defensins that are responsible for crystallizing the solution and piercing the pathogen membranes. And so neutrophils are also responsible to aid in this process. So then the toxic, cytotoxic chemicals within our pathogen or our phagocytes can destroy that pathogenic cell wall and the contents of it, and so then we can release that material out of the body, and so it will not cause us any more harm. So when we look here for the steps of phagocytosis, so here step 1, the microbe is going to adhere to the phagocyte, and then via endocytosis then we're going to have ingestion of that microbe, and then there's going to have that phagosome, which again, that phagosome is going to be the binding of the lysosome to that bacteria, and so a phagolysosome will form, and lysosomes tend to have an acidic environment where it's going to puncture the wall of that bacteria, and then we'll have different enzymes destroying it, and then so via exocytosis, then all that has turned into cellular debris that our body can get rid of. Another mechanism by which a different type of cell can destroy different types of pathogens are through natural killer cells, or these are sometimes called cytotoxic T lymphocytes as well. Okay, so their function is to destroy body cells that have any type of foreign molecule. So these are a little bit different because these are non-specific for non-self, and so they're able to attack virus or cancer cells without antigen recognition. So these are not phagocytic, where they must come in contact with their victim cells, and what they do is they release these different types of proteins called perforins that perforate the target cell plasma membrane. And so this also then they're able to induce apoptotic enzymes, which are called caspases, and so that triggers the death of those foreign cells. So there's two main types of cell or things or targets that these cells are looking for. So the first are going to be virus-infected cells, and the second, cancer-infected cells. So they're going to attack and kill these target cells directly after they bind to them. So no phagocytosis with these types of cells. Inflammation is going to be our local response to some type of injury or infection, and so it's going to activate a lot of those cells that we've been talking about. So the main function of inflammation is to destroy or inactivate foreign invaders and to set the stage up for tissue repair. So inflammation is triggered by different types of injury, and this includes trauma, different types of heat injury, chemical radiation, infections, among many other types. And so the benefits of having an inflammatory response is that it helps to prevent the spread of injury, to help dispose of cellular debris and pathogens, and mostly these are going to be phagocytes that act as the key players. And again, those were the neutrophils and macrophages. And it's also going to help promote tissue repair. All right, so then with the inflammatory response, we see different signs and that are associated with vasodilation. And so dilation of those blood vessels primarily are going to be by histamine again. And also there's different prostaglandins that induce that redness and heat. We have an increase in permeability with edema, and so this helps to increase the pressure. And also we'll have pain that's associated with the different types of toxins, and primarily done by that caspasin. So some of the inflammatory mediators that are released by these injured cells, again, include histamine. And so histamine is responsible for that vasodilation and also increase in vessel permeability. Also, we'll have different types of prostaglandins, which also help with vasodilation. But these also are in part for why we have induction of the fever, and also helps with the triggering of that pain sensation. Also, we'll have cytokines, and these are going to be different chemo attractions for leukocytes to come to the local area. And one other one that's a big part is our kinins, and these are going to be important for dilation of those blood vessels. So with edema, we have the dilution of different harmful substances, and so these are due to antimicrobial mediators that are found that are actually able to penetrate into these affected tissue areas. Also, we'll have increase in the nutrients and oxygen that are able to go to that local site of injury, and then also there's an entrance for those clotting proteins that are able to enter the area. And again, with edema, this was due to an increase in vasodilation and increase in vessel permeability, where we'll have an increase in plasma to the tissue space. And another site that we see are epithelial breaches also stimulate beta defensins, and these are going to be antimicrobial peptides that we find within epithelial cells to prevent further penetration of bacteria or different pathogens. And so here is a flow chart of the many different and this is actually simplified of a flowchart of what is happening when we have some type of tissue injury. And so the big thing, what you should focus on, again, are these responses in red. So with heat and redness, pain and swelling, these are all going to be the common events that occur with any type of inflammation. So the mechanisms by which these cells work during an inflammatory response to induce all those different changes include phagocyte mobilization, and so this is where those phagocytes are able to move into those damaged areas by different neutrophils and macrophages. And again, neutrophils are going to be the body's primary defense against bacterial pathogens, okay? So then one other process that we have is leukocytosis, which is an elevation in the total white blood cell count due to the increase in neutrophils. And so this is for rapid release of those white blood cells that come from bone marrow. And then also we'll have marginization, where we'll have an increased vascular permeability, and so this will decrease the fluid in the blood vessels, again causing a little bit of edema in certain areas because that blood plasma has exited to the site of injury. And so we'll have slowing of blood that's going to flow, and then neutrophils then are able to move to the vessel margins, and we'll have different cell adhesion molecules, or CAMs, that allow the neutrophils to cling to those vessel walls so that they can rebuild the broken or injured tissue area. And so this is termed as pavementing. After margination, then we have diapedesis, and this is where neutrophils can migrate into the capillary walls, and this can occur within minutes after an inflammatory response, where these neutrophils literally squeeze through endothelial cells in the vessel walls, and neutrophils are going to be the first to respond to any type of injury or injury, yes. And so then what happens are chemotaxis occurs, and this is where inflammatory chemicals are going to attract more neutrophils to come, and then creating a high concentration gradient for those neutrophils to those certain areas. After some time, monocytes then will also undergo this process of diapedesis and chemotaxis. However, it's neutrophils that will go through this first. And so what we have here are neutrophils are going to enter the blood, and then we'll have the margination, where they're going to embed within that capillary wall, and then they're going to squeeze out between those spaces in the cells with diapedesis, and then they're going to release their different chemotaxis and different chemicals to have those different inflammatory responses come. And again, so this happens with neutrophils first, and then it will happen with those macrophages. So then one of the last steps is that the macrophages are going to clean up the cellular debris and pathogens where they're going to ingest this material. And so if pathogens were associated with the injury, activation of a complement cascade occurs. This will activate then when antibodies bind to molecular targets or antigens. And so this will further promote phagocytosis, lysis of these targeted cells, and even more inflammation to continue and occur. So we've been mostly talking about different pathogens of bacteria and fungi, but viruses are a little bit different because they release their nucleic acid, either of DNA or RNA, into the cell cytoplasm, and then that information on the nucleic acid then is incorporated into the cell's DNA, and so can adjust and to produce more of the viral DNA and different cellular components and organelles. And so instead of the cell making what it normally does, now it's making material for that virus and releasing that from the cell. which sometimes can be deadly for the cell. So there's a mechanism called with interferons. They're responsible for creating these antiviral proteins. These interferons produce a nonspecific, short-acting resistance to viral infections and are produced by almost all cells in the body in response to microbial infections too. How interferons work is that they inhibit viral replication. Even though the virus can penetrate the cell, it slows it down, and so the virus isn't able to reproduce within the cell. Also, interferons can induce natural killer cells, and so that also helps to get rid of those pathogens. So far, the FDA has approved treatment with interferons for hepatitis B and C, for genital warts, and for multiple sclerosis. And so in this image, what we have here is the virus is going to enter the cell here, and usually what happens is that it's going to continue on and infect the nucleus so that it can change the DNA, mRNA, and then to make different types of proteins. However, the interferon gene is turned on, and so what happens is that it inhibits the molecule from being produced. And so with the production of now antibodies that are found, this interferon then can bind to it and then can block the production of more viruses, so that stops that whole process.